Hi guys, today we are going to revise this topic of microbiology for your upcoming Neet PG exams. This is the topic I have selected for today, and this is about gram-based lab algorithm. We are going to study about gram-positive organisms and how they are classified, and the antibiotics onto which they are either sensitive or resistant. So here we begin. First of all, gram-positive bacteria are those uh, which give the purple color under microscope, and these can be either of three shapes. These can either be cocci or round, these can be bacilli or rod shaped, or these can be branching filaments, as you can see on the screen. First of all, cocci, which are the round gram positive organisms which show purple onto the microscope, because we all know that gram negative organisms show red or pink onto the microscope. First of all, it is showing that these are anaerobic and facultative. Facultative are those organisms which are able to survive either in the presence of oxygen or in the absence of oxygen. So for their further classification, we will need to do a catalase test. Catalase is the enzyme which enables the bacteria to break down the hydrogen peroxide into oxygen and water. So you can see the white bubbles forming in the slide. So if they are catalase positive, this will shift our paradigm to staphylococci. We all know that streptococci are gram-negative catalase negative organisms. We are talking about gram-positive organisms in here. While we know that these are staphylococci, we can further go on to coagulase test. Coagulase test, this further divides them into Staph aureus. Okay, if the coagulase test comes positive, coagulase is the enzyme which enables the bacteria to dissolve the blood clots. If these can move into the blood clots, that helps the bacteria to spread. Wherever clot is found, bacteria can cross that clot, and it helps in the spreadability of the bacteria. Thus, Staph aureus, it has that ability. And if it is coagulase comes negative in the catalase positive category, then we will either go for novobiocin sensitivity. Novobiocin is an antibiotic. If it comes positive, then we can be sure shot that this is Staphylococcus epidermidis. Epidermidis is found onto the normal skin, and it is normal flora of the human body. If it moves across that skin and moves onto the endocardium in the heart, it can cause endocarditis too. So we know that this is prevalent in intravenous drug abusers. If the novobiocin sensitivity comes negative, which means that the organism is novobiocin resistant, we can sure shot know that this is Staphylococcus saprophyticus. Then we will move on to the next category, which is Streptococcus. These are catalase negative. We can further classify them on the basis of hemolysis. Chocolate egg is used. On the hemolysis, we can see three types of hemolysis. Partial hemolysis, which is green, you can show on the screen. You can see on the screen. Beta hemolysis is complete hemolysis, wherein the blood agar completely becomes clear. If there is no hemolysis, then we can classify them into the gamma category. Alpha category, partial hemolysis; beta category, complete hemolysis; and gamma category, no hemolysis. We need to understand that in gamma category, these organisms grow in bile and in NaCl with a concentration of 6.5%. So you can see these are organisms that can grow in NaCl as well as in bile. And these are group D enterococci. Okay, we will move on to the next category. This is partial hemolysis or alpha category. Next we will do optogene sensitivity and bile solubility test. If the optogene solubility comes positive, this is strep pneumoniae. We know this causes the lobar pneumonia, most common cause, and it is an encapsulated organism. If optogene resistant is the organism, then we will place it into viridans streptococci. This is the organism. It does not have any capsule. and the organism is step mutants. Okay, what have we just studied? That in partial hemolysis, aptogen test will be done, and if it comes aptogen sensitive, then it is strep pneumoniae, and if it is aptogen resistant, then this is viridian streptococci. It is a more normal flora of human mouth and present predominantly in teeth and in oral cavity. For the complete hemolysis test, we will do bacitracin. Okay, you will know from this that bacitracin is the starting with B and beta category shows the complete hemolysis. You can correlate them. If bacitracin comes sensitive, then this is group A, step pyogenes. It causes the post-streptococcal glomerulonephritis or rheumatic fever to rheumatic fever. And bacitracin comes resistant, then this is step age elective. Alpha hemolysis, you can see the shape of alpha, which is also like the shape O, alphabet O, so you can correlate them too. And for the last category, we know this is Enterococcus or non-enterococcus, such as step bovis. If you like it, please give a thumbs up. We'll see you in the next session.